Let's continue our worked problems by going through some pedigrees. So just as a review, or just in case this isn't in the video lecture, let's talk about what a pedigree is. So a pedigree is essentially a family tree that's used to study patterns of inheritance. So it follows a particular trait through a family. Now, this might be a disease trait, or it might not be. It could just be a trait of some sort of physical characteristic. So males are square on a pedigree. So anywhere you see anywhere you see a square, it's going to be a male. Females are circles. So anywhere you see a circle, that's female. Affected individuals have their circle or square shaded. That tells you that they have the trait. A bar connecting two individuals means that they had children together, and coming down from them is going to be their offspring. Now, by convention, the offspring are written in birth order, left to right, firstborn being the oldest. So this is often used to determine the type of inheritance pattern of a trait. So this might be dominant or recessive. And then you also have to determine if it is autosomal or sex-linked, which means it is found on the X chromosome. So for every pedigree that you are given, you are going to be asked to determine if it's dominant or recessive and if it is autosomal or sex-linked. So throughout this video, we'll talk about all of those. So let's look at dominant traits first. So I'm going to say one thing here, and then I'm going to tell you I don't ever want to hear you say it to me again. Okay, the easiest way to determine that something is dominant is that it appears in every generation. But recessive traits can also appear in every generation. So you may not use that as an example of, or a support for your argument that it is a dominant trait. There are a couple of other ways to determine that something is dominant. So you can start by noticing that this one occurs in every generation, but do not tell me that on a test. All right, so related to that is that every affected child has an affected parent. Now you might say, oh, but that means it appears in every generation. It does in most cases, but not every affected parent is going to produce an affected child. If this family here, this bottom one, didn't have this child, they only had this one, which is totally possible, you don't see it in every generation. So it is still dominant, but you don't see it there. So every affected child has an affected parent. That is one of the ways that you can determine it. The most telling way to determine that this is dominant is this situation here. Two affected parents produce both affected and unaffected children because both of those parents are hiding the allele. This one might not appear in every pedigree that you see, so you have to look at the different ways that you can determine that it is dominant. So look for this one first. Do two affected parents produce both affected and unaffected? If yes, your job is done. Otherwise, you have to look. Does every affected child also have at least one affected parent? Again, that one does not always rule out recessive, okay? So this crossing heterozygotes, though, produces both affected and unaffected. Look for that one first. And if you see that one, you know that you've got a dominant. So once you know that this is dominant, you can assign all of these unaffected people a homozygous recessive trait and then work backwards. So if these people are affected, you know that they have to have at least one big W. This person and this person had to both pass that allele to this individual. So you know that both of the parents have that allele. recessive traits. The easiest way to determine this is by looking for unaffected parents that produce an affected child. I always start with this one because this is the easiest one to definitively say, yes, this is recessive. If you see this, it's recessive. If you don't see this, it is probably dominant because both parents are masking that allele. Those parents are called carriers because they have the trait, but they are not expressing it. Now, this often skips a generation, but it doesn't have to, okay? If this individual marries someone who is heterozygous, they can have a child that is homozygous. And then we see it does not skip generations. Grandpa, mom, daughter all have it. So skipping a generation does not make it recessive, nor does it rule out dominance, okay? So, not that if it skips a generation, it is likely going to be recessive, but just because it does not skip a generation doesn't make it dominant. Hopefully that makes sense. Again, once you determine that this is recessive, then every affected individual is going to be homozygous recessive. So you can then assign all of these affected individuals homozygous traits, and then you can work backwards. This person is homozygous. Both of the parents had to pass on that allele. If they are unaffected, they are heterozygous. This individual can be either one, and because her parents can produce big T or big T little t. So you don't know what she is unless you have information on her parents. All right, so let's look at a couple of these. Let's start with this top one. So remember I said to look for the situation where two unaffected parents produce an affected individual. Do you see that there? You should not see that there. That does not appear in this one. So let's move to dominant. Do you see a situation where two affected parents produce both affected and unaffected children? We don't see that one here either. So now we need to think about, does this skip a generation or do we see it in every generation? Well, we have 3 generations here. We have grandpa, mom, and daughter. So we see that it is appearing in every generation. And also every affected child has an affected parent. So daughter, mother, mother, grandfather, son, father, daughter, father. So this one is going to be dominant. Okay, so everybody that is dominant is going to have at least one dominant allele. We'll fill in that other allele in just a minute. Keep wanting to jump ahead. All right, that means everybody else is going to be homozygous recessive. Now, at the risk of confusing you, the most common thing that I hear students say is, ooh, that one is recessive because it is affected. I don't know why students assume that, but it is not true. Okay, the shading in of the trait does not imply or make you assume that it is recessive. Also, by being infrequent or occurring in a small number of individuals does not make it recessive. Occurring more frequently does not make it dominant. Those are very common mistakes that students tend to make. So let's go through and finish filling out our genotypes. So this parent produces both let's not go there. This individual has unaffected children that are little d. That means one of those D's came from mom, the other had to come from dad. This individual here, we don't know. Oh, yes, we do. The big D came from dad, so the little d has to come from mom because that's the only thing she can pass on. Same thing over here. Dad passed on the big D, so mom had to pass on the little d. And then same thing here, because dad is homozygous, he had to pass on that allele. All right, let's look at pedigree B. So let's look for this one first. 2 unaffected parents produce an affected child. Do we see that? We do. 2 unaffected parents produce affected children. Okay, that means that these parents are hiding it in the heterozygous. So these guys are heterozygous. These guys are affected. All of these other individuals are we can now label their traits. Let's start down here with this individual here because this is interesting. She has a big D. We know that because she's unaffected. All of these other individuals are also unaffected, so they all have a big D. Dad had to give a little d. So we know that all of these individuals got a D from dad. Little d from dad. Do we know what mom's allele is? We actually do not. She could be big D, or big D, little d. However, since none of the children are little d, which you would expect 50% of them, it is more likely that she is homozygous dominant. Having only 3 offspring, there's a chance that, out of 50%, you can't have 1.5 that are recessive. But considering that they made 3 individuals that all display the dominant trait, it's most likely that she's big D. Down here, this guy could be big D, little d. Same thing with these guys. We don't really know for sure without some more information. So again, everybody else has at least one big D. This looks like it's a rare trait. So it is most likely that most of these people, well, I don't know that I really want to say that. We know that this parent had to get a D, little d from somewhere because they produce a child. So one of these parents is going to have to be big D, little d. The other is going to be big D, little d, or big D, little d. This individual is either big D, or big D, little d. So I'll let you work out the other ones. If you don't know, just make sure that you write both big D, or big D, little d. This one here, we're going to start by looking for 2 unaffected parents with an affected child. We see that in this generation with this child and this generation with this child. We see that in all of those locations. So this one is also recessive. Now we're going to come back to that one in a minute. All right, so let's move on to incomplete dominance. Remember that incomplete dominance is where you get an intermediate phenotype in the heterozygous individual. So let's say we have a bird, and that bird can be, let's say feather color can be blue, feather color can be white, or in the heterozygous individual with an intermediate phenotype, we get silver. Okay, so a bird that is blue is going to be homozygous for the blue allele. A bird that is white is going to be homozygous for the white allele. And a bird that is silver is going to be heterozygous. So on an exam or in a homework assignment, you may be asked to determine if this is incomplete dominance or codominance or regular dominance. So you have to be able to look at the situation and recognize that there's an intermediate phenotype. All right, so let's first cross a blue bird with a white bird. all of the offspring are going to be heterozygous, which means they are all going to be silver. So all silver, all heterozygous. All right, let's take one of these, our silver bird, and let's cross it with a white bird and see what happens. All right, so we are going to get, heterozygous. Heterozygous, so these are going to be silver. And then homozygous, so these are going to be white. So we have 2 heterozygous to 2 homozygous, which is going to be 2 silver to 2 white. Now you probably noticed that this is a different notation here. That is to indicate that this is a different type of dominance. So incomplete dominance is denoted by a capital letter. In this case, I chose F for feather color, and then the allele is an uppercase. So I chose B for blue or F for white. For codominance, we have the same situation. This is where the heterozygous, or a similar situation, I shouldn't say the same. A heterozygous individual displays both phenotypes together, not an intermediate, but distinctively shows both. So this cow, let's look at cattle color. So let's say that the red color is indicated by an R allele and white color is indicated by a W allele. Heterozygous individuals are going to be spotted red with white. See how we can see both the red phenotype and the white phenotype, as opposed to, say, a lighter red or like a orangish color. Okay, if it was lighter red all over or orange all over, it would be incomplete. But because we see both the red and the white on this cow, it is codominant. You might see codominance written this way. This is the way that I tend to write it, indicating using the capital alleles for dominance, indicating that they are both showing their dominance. You may also see it written like incomplete dominance, where let's say it's coat color red and coat color white. You might see it either way. I don't care as long as you can clearly explain to me in the phenotypes what is happening. So let's cross a spotted cow with a red cow. So we cross spotted with red, we get red, red, or spotted. So we get 2 homozygous to 2 heterozygous, or 2 red to 2 spotted. Let's cross a heterozygous with a heterozygous. So let's write that over here. Heterozygous with heterozygous. Here we're going to get red, spotted, spotted, and white. So here we get a 1 to 2 to 1 genotype ratio. We get a 1 red to 2 spotted to 1 white phenotype ratio. Again, notice that the heterozygous displays both of them equally. So this cow would be the heterozygous. All right, multiple alleles. So we're going to do this in lab, but I will do a couple of quick examples for you. So if you are type A blood, type A blood, you can have the allele combination IA with a recessive, or a homozygous A allele. Now the reason is we can have homozygous individual that's showing the A trait, or we can have an individual with the recessive O allele that is showing the heterozygous showing the A blood type. B is going to be similar. So A or B is dominant over O. O blood type is going to be the recessive. The fourth one, the AB blood type, is going to be codominant. So we have both codominance and simple dominance in our blood types, okay? So you have to understand that your ABs can be heterozygous, your O is homozygous, and your AB is heterozygous. So I'm just going to do one example here. Let's cross an individual who is AB with a heterozygous A. So your type AB cross a heterozygous A. We get an A blood type, an AB, an A, and a B. So for this cross, we can get one type A, two type A's, because these are both type A's, two type A's, one AB, or one B. We're going to do this again in lab, so that's all the examples I'm going to do here. But if you have questions about it, please let me know. All right, so let's do some X-linked trait examples. So this is an example showing, we'll just say that this is colorblind allele. Now remember, in X-linked traits, the Y chromosome does not have a gene. So if you look over here, this gamete is only going to be Y, okay? That sperm, we're seeing that down here, that sperm is only going to be a Y. There is no colorblind allele. That is the most common mistake that students make, is attempting to put an allele on the Y chromosome. The Y chromosome does not have a colorblind gene. Remember, the Y chromosome only has genes that are related to testes development in males, or the formation of male genitalia. The X chromosome, however, has that allele. So females are heterozygous. Males are what we call hemizygous. Now, I don't necessarily need you to remember that word, excuse me, hemizygous. It just means that they have only one copy of the genes on the X. Okay, so keep that in mind. That's important. That is why males and females show different ratios for these traits. So if we cross a female who is homozygous dominant with an affected male, this diagram shows sperm and eggs. So the eggs, the possible eggs, are going to be X with a dominant allele. Sperm are either going to be X with the recessive or Y. So when we cross this, we get heterozygous females. These are both normal. So we get two normal females, one normal male, and two normal males. I looked at that wrong. I got ahead of myself. So two normal males, two normal females. Notice that I paid attention to saying whether they were male or female. If you just tell me 50% affected on a sex-linked trait, your answer is wrong, because with a sex-linked trait, we need to distinguish between males and females. Okay? Unless the question specifically asks what percentage of offspring are affected. But I very rarely ask that question because I want you to tell me the difference between males and females. All right, so let's do a couple examples of X-linked. So the page prior, we did this one, a normal female with an affected male. Let's do a heterozygous female with a normal male. Okay, females, inheritance with sex-linked genes behaves exactly as Mendelian inheritance. You need two copies to have a recessive gene. With males, you only need one copy. That makes sex-linked or X-linked disorders more common in males, but can happen in females because females just need two copies. So if they get an affected allele from their father and from their mother, they're going to be, they are going to be affected. So more common in males, but can affect females. So let's do our cross here. So for this one, we are going to get a normal female, So we have two normal females to one normal male to one affected male. So this is going to be a colorblind male. All the other individuals, including all the females, are normal. Now let's say that we have a heterozygous female with an affected male. All right, so female's gametes, male gametes, normal but carrier female, affected female. So there we have our affected female, normal male, affected male. So in this case, 50% of the children are affected. So we have one normal female to one affected female to one normal male to one affected male. Now don't forget about those genotype ratios. I am not writing them out to save time, but don't forget that I might ask you for those genotype ratios. All right, so let's look at an X-linked pedigree. Remember I said we were going to come back to one. I put that one here on the right. In an X-linked pedigree, you tend to have all affected males. Not all males are affected, but all individuals who are affected are males, or far more males than females. So you might have this situation where most of the affected individuals are males with one affected female. Remember, females can have these traits, but it is much less likely because they have to acquire two copies, whereas males only need one. So the way to determine a likely X-linked gene is by one, it being recessive, so two unaffected parents produce an affected child, and two, all of the affected individuals are male, or the majority of them are male. Now this is because the mother is a carrier and she is passing it on to her sons. Okay, dad cannot pass an X-linked gene on to their son. Mom can pass it to a daughter, so she might be a carrier. I haven't done this, so I'm just making notes at the moment. She might pass this gene on to her daughter. Okay, so let's actually figure out the genotypes for these guys. So X-linked recessive, all affected individuals are males. So let's call these guys affected. We'll use the H allele. They had to get it from their mother, so their mother has to be a heterozygous carrier. Dad is not affected. Remember, he only has one copy of that X chromosome, so if he doesn't have the disease, he's dominant. Same thing with this dad here, this dad here, this dad here, and this son here. Mom, for this one, we don't really know. She could be either one. She's an outsider marrying in. All right, so let's look. This individual had to get this allele from his mother. She's not affected, so that makes her heterozygous. This male here is affected. That means he had to get that allele from his mother who was a carrier. This individual here had to get the allele from his mother, who was an affected carrier. That means that she probably got this allele from her mother. So she probably got the recessive allele from her mother and the dominant allele from her father. On the left here is another example of an X-linked pedigree, only this time it shows the carriers. So the carriers are indicated by this half-colored circle. Sometimes they are indicated by the half circle, sometimes they are indicated by a dot in the center. It just depends on how they make their pedigree, so look out for either one of those. You'll notice lots of females are carriers, affected male, etc., etc. Here there is one affected female. It does not mean it's not an X-linked, because the majority of the affected individuals are males. So out of this whole big family, there's only one affected female.